Hi, my name is Andrew Kurt, and this is the Unit 3 presentation on the paper The Genomic Code, a preservation and coding molding of chromatin structures and a solution of the non-coding DNA mystery. Um, what is the non-coding DNA mystery? The non-coding DNA mystery, as the paper explains, is that around 80 to 95% of the total genome is for non-coding DNA, that they don't code for any genes. And the mystery was, what is the purpose of all this non-coding DNA? Why is 80 to 95% of our DNA essentially not doing anything? And there were theories that it was just junk DNA or selfish DNA that was essentially transposing itself. But what the paper goes on to explain is that the non-coding DNA can actually help fold the overall 3D structure of the genome. And the evidence they give to that is when looking at long strands of DNA sequences that are like 100,000 base pairs long, you can see unique 3D structures. And these structures are called isocores. And the isocores can fall into different families that are defined by the GC content in these long strains of DNA. The isocores fall into two versions: LADs and TADs. The LADs are defined by having poor GC content and lots of A's and T's. And it's these A's and T's that give them their unique 3D structure. They cause local stiffness and bending of the genome, and this causes association with the nuclear lamina. That's why they're called LADs, lamina associating domains, while the areas that have high GC contents are called TADs, or topographical associating domains, and that they form these 3D structures that force the DNA, that extrude long portions of the DNA into peaks. So these LADs and TADs are then further associated with other characteristics of the DNA. In the TADs, the topographically associating ones, that they have high GC contents, and these TADs and high GC content areas are also in areas that are gene rich, and they have a lot of genes, and they're also associated with open chromatin structure. And so while the LADs, one with high A's, are associated with the gene poor regions and don't have those extruding features. And so what they theorize is that the 3D structure that the GC content creates, these long extrusions in the DNA, that these help the genes, get transcribed properly, and help with transcription by making them more open and available to all the proteins and enzymes that are in the nucleus. He further goes on to explain that it's the LADs and TADs that are responsible for the 3D structures, and not the nucleosomes, as it was thought. And the evidence he has for this is that when the nucleosomes are not attached to the DNA in DNA replication, these long-range 3D structures are still there. And when the DNA replicates, the 3D structures show up before the nucleosomes are then attached to the DNA. And so that's essentially the DNA code itself which creates these structures and keeps these structures going. And so overall, I thought that the paper was very interesting. I never heard of the DNA composition itself and coding for the 3D structures of it. I always thought it was the proteins that attach to these non-coding regions that did that. So it's interesting to hear about it's interesting to hear about how the structure itself can, or the composition itself can cause these structures. Um, However, I did notice that the paper says to see that this discovery has solved the mystery of non-coding DNA functions, and I think that maybe solved is too high of a word. That there are still some more questions about it, and more questions, more answers that can show up. Like it said that the in the TADs they're extruded by using cohesion binding to give those like peaks and really bring out the DNA. But when you take cohesion away in lab, it doesn't cause any noticeable effect to transcription. So I feel like there's more to learn about that the if they think that those help transcription and help and since they're associated with open chromatin and transcription in genes, that even if the cohesions aren't there and they're not formed as well, that transcription isn't really affected as much. I think so. I think there's still more questions that need to be answered about this, but overall, I thought it was interesting and informative, and I enjoyed reading it. Thank you.